What I wanted to do in my talk is give you a couple of inspirations from our work into insect brains and how we can interpret what we see in the insects for building different types of AI. And I wanted to start with the observation that, of course, lots of the common AI that we are looking at the moment is based on deep learning, where we have networks that look a bit like this. This is a VGG16, by now almost an old technology. But what you can see is each of these little plates here represent a layer, and you have these deep architectures where you have several of these layers with literally thousands to millions of units in them that then make a computation, and they break it down in very fine steps, so to speak, and do it in these many layers. Whereas if you look at an insect brain, there's no way that such a deep architecture could be situated in the insect brain. So if you just, for instance, take the olfactory system, it starts with the antenna lobe. Well, of course, first there's the antenna with the sensory neurons, then the antenna lobe here in the bluish color. And then we can get to the calyx of the mushroom bodies, the lobes of the mushroom bodies. So that's maybe 3 layers. And from there, one is already in kind of premotor area. So in essence, in the insect brain, and I would even argue in mammalian and human brains, there's no space for such deep architectures, even though there has been some recent work on similarities between human brains and deep learning architectures, where the argument was that several of those layers might be implemented in a single layer of cortex, for example. But for the insect brains, for sure, it's not going to be that deep. So how do the insects cope without being able to do deep learning? They cope well. So if we look at what insects can do, the list is long, and I've just picked out a very few things here about bees and moths. Bees can memorize routes over long distances and reliably return to their home. Bees and ants can do path integration in the absence of learned routes. Bees can communicate food locations with the waggle dance you might have heard of. Then moving on to moths, moths can find their mates from more than a mile away, and they can separate antagonist odor sources that are only millimeters apart, which is a very difficult technical problem. And so on, the list goes on. So how do they do it? And I just wanted to show here one example where we had taken inspiration from the architecture of the insect brain. It showed that it can solve quite general computing problems to a certain level and with certain properties. So the example that I've picked here is a relatively old work from our lab, where we looked at insect olfaction, and we built a model of the insect olfactory pathway that I just pointed out on the bee brain there. So we have here the equivalent of the antenna lobe. And what we used as the machine learning benchmark task was the recognition of the MNIST digits. People who work a little bit in machine learning will recognize this as a very standard benchmark that by now is considered as long solved in machine learning. But I'm coming to my main point in a moment. Yes, I'm not just giving you some history here. But the task is to recognize handwritten digits like this number 5 here. And the anatomy of the olfactory system is then that we have the antenna lobe, then we have what we believe are random projections to the mushroom body calyx that has the canyon cells in it, and there's lots more canyon cells than antenna lobe input neurons, and the connections are presumed to be random. And then we have plastic synapses to the output regions of the mushroom body. And here we have just located like 10 neurons here to signify the final decision of the system. So you see a very shallow system with random connections that don't learn, and just a single layer of learning here with a Hebbian type plasticity. So with that system, we could show that we can learn this task to a certain level of performance. And so here you have a plot where I show the final performance after learning against the number of canyon cells in the mushroom body. And then making the point that with a relatively small system, I made the arrow here to the number 1,000, yeah, with a relatively small system, we can already learn this task to more than 80% accuracy. Now, I said earlier, this isn't in the AI business, this isn't considered a difficult problem anymore. And AI formulations with artificial neural networks, semi-deep networks, let's say, can solve this task to 99%. So a lot better than this model. But on the other hand, it is very resource unintensive, because we just need these 1,000 cells, and it's not displayed here, but they're also only very rarely active, and this is a very sparse activity and connectivity, which means we don't need much energy to run this either, and we can do it to more than 80% accuracy. In addition to that, we found that it's also very robust. So if I start with a few more cells, and this is a plot where I show the performance, how it degrades if I remove cells, and I started with the number of cells being 50,000, so that's this point here. And you can see you can remove about 99% of the cells before you start seeing real impacts on the performance. And that, interestingly, is the same performance as if you had started with that few cells. So if you just think about it for a moment, this is after training, I remove the cells, I destroy part of the system, a large part of the system, and it keeps performing very well until the point where, if it had been a smaller system of that size and I had trained it as the smaller system, that would be the same performance. So that is something you can't do with a deep network. If you start destroying deep networks, they start failing much earlier. Okay, so there's a bit of inspiration there. So it's not performing maybe as well as a huge deep network, but it's efficient and robust in what it does. Okay, since then, lots of things have happened. So that work was from 2004 and 2009. We have done some follow-up works. But there's also a parallel development of so-called extreme learning machines that are using the exact same inspiration, coming, but as far as I can tell, completely independently from insects. But there's also been more recent works looking at these aspects again from 2014-17 here from some well-known labs. So that's one type of inspiration we take from insects, where maybe there's somewhat simple and somewhat less performant, but very robust and efficient algorithms that we can learn from the insect brain. The other aspect I wanted to talk about is, again, now properly in olfaction, and that is about solving problems in a non-standard way. And that is here, my example is the separation of two odor sources. And here's a nice image of how two odor sources would emanate in the environment in complex plumes. So unlike one might think naively, odors in the environment don't disseminate in nice gradients and smooth concentration profiles, but they disseminate in these messy plumes. This, however, is rather than a disadvantage, is an advantage for being able to separate odor sources, because as you can appreciate from this image, blue and red actually only rarely come really together to form a different color, but they're only intermingled, and you get intermittently some red and some blue, which means from the temporal structure of the plume, you can tell that this is two odor sources rather than one. If it was from one odor source, we would have kind of an intermediate color in all of the filaments, and it would be fully mixed. So we know that insects use this information, and this is some behavioral work from our collaborators, Paul Siska et al. From 2012, where they have shown that if you train a bee to respond to an odor A, and then you test it with different mixtures of this odor A with an odor B, and the difference between the mixtures is that it's either fully mixed and two odors come at the same time, or you mix it with a nice olfactometer in a way that one odor comes a bit earlier, the trained odor here in the first example, or the non-trained odor in the second example comes a bit earlier, and then the other odor is shown. And this is to scale, so this is done with a tiny delay of 6 milliseconds. And what you find is that in training, you can train the bees to respond to the odor A. And if you then take all the respondent bees, so we renormalizing here the axis a little bit, and test them on these mixtures, you see that they significantly more respond to the asynchronous mixtures here with the hatch than to the synchronous one. So in essence, they can use this information of asynchronous arrival of odors to see whether it's a mixture coming from two sources or from one. Now, how do they do it? Our hypothesis is that they just simply use inhibition between the two odor channels to decide. And so here's an illustration of that. So if you have a plume arriving and the two odors here in purple and in green, they are completely synchronous. So it's fully mixed mixture at the source and just a single source. Then, of course, they come at the same time, which means if two receptor neurons are activated by these two orders, they would, if they inhibit each other, they would reduce the signal coming through. But if they come at different times, then they wouldn't be active at the same time, and there would be much less inhibition. So you have a much larger response overall. It looks all messy, but it's much larger here than here. You can probably appreciate that from this example. And then we have investigated in circuit models how that might be done in the brain. And there's an older paper here where we show it could be done in the antenna lobe, where if there's different order channels here with different receptor neuron types, that they could inhibit each other through local interneurons in the antenna lobe. Here's the first stage of information processing in the brain. But more recently, we have seen work that has shown that there's ephaptic interactions already between the sensory neurons and the sensilla. And we have shown in some computational work that's just coming out that this can equally well do the job of distinguishing between the synchronous and asynchronous mixtures as this network in the antenna lobe. And actually, when they work together, they seem to be working best. And now this is a curious thing. And this is, I'm almost there at the end. This is something that you normally would avoid. If you had two information channels, you would avoid it like the pest to mingle them up together and mess up your signals, right? You would want that you get nice and clear signals that you can then process in the brain. But here it seems to be that nature has chosen, or evolution has evolved, a method where you kind of destroy your nice signals and you represent the information in the world suboptimally, as it seems, to achieve a certain purpose. Now, that is an inspiration, I think, that's not so commonly seen from machine learners. And I wanted to just demonstrate that this is not some insect-specific thing, and I want to end on this, but it's something that you do as well. Yeah? Your brain does this all the time. Actually, it's doing it right now while I'm talking to you. And here's a demonstration of that. I can't do it with odors because I haven't figured out how to transmit odors through the internet yet. And there has been some work into electronic stimulation of odors on TVs and things, but it hasn't gone anywhere. So I'm not going to do it with odors, but I'm going to do it with tones. So we're going into the domain of hearing here instead. And what I have here is two examples that I will play to you of tones that are a mixture of 20 pure tones that are not harmonious. And you have one mixture where all the tones come on at the same time. And you have one mixture where you have lots of tones coming on at 0 and one single tone comes on at 160 milliseconds. And then let's see whether you can hear a difference. So try to concentrate and see whether you can hear, whether you can see a difference between these two. So I'll try this first. So I hope you could hear the difference. Normally with a physical audience, I would ask for hand signs and things, but I trust most people hear this quite well. And most people describe it as that in the second example, you hear a second high-pitched tone coming on top of the original tone. So the tone seems the same at the beginning, and then there's a second additional high-pitched tone. And that's an interesting thing. So if you think it through, maybe you already caught on to it, but for me, it took a while until it actually dawned on me that this is a very interesting thing. Because in the end of the day, if you wanted to represent physical reality optimally in our brains and in our perception, what we should be hearing is the same tone at the end, right? That's where they're physically the same. And we should hear different tones at the beginning, and then we would have an optimal representation of what's going on in the world. But we're doing the opposite, yeah? So the tone first sounds the same because we don't notice the missing frequency. And then we add perceptually a second sound source, a second tone into it, even though actually the physical signal is then the same as this one. So clearly the brain and our sensory systems are not trying to do anything optimal or particularly faithful. They're doing something that's useful. And that's something I think we can learn even from simple insects. So to summarize it up, I think what I've demonstrated today is that there are non-standard kind of methods, things that we don't think how it should be done in terms of signal processing that brains do to process information and lead to successful behaviors. And they encode only what's useful and leads to survival, and not necessarily what's the most information, the best information, or the truest information. about the environment. And often algorithms are only good enough and not necessarily optimal. And with that, I would like to acknowledge my collaborators over the years and you for your attention. Thank you very much.
Thank you so much, Thomas. That was very interesting. I could hear the difference between the beeps.
Perfect.
Sorry, I've just turned my video off. There we are. So we do have one question so far, and to the rest of the audience, please do remember, feel free to drop a question in the Q&A. The question so far is from Richard Barrett Jolie, who says, dropping neurons in deep networks, maybe you can't lose layers, but we do use dropout layers to remove neurons, do we not?
Yeah, it's right that one uses dropout to temporarily remove neurons to make the system more robust. And arguably, that would mean one could also remove them permanently without too much detriment to the overall result. But there is a limitation how much one can do that. Arguably, here is a limitation as well. Of course, I can't remove the entire mushroom body. This is a question of shades of gray here. Now, is it some more or some less? And I would argue if you have these sparse codes with a lot of inactive neurons, like in the mushroom body, you can remove more than in a deep neural network where all units normally contribute to the computation at all times.
Thank you. I have a question that's maybe facetious, but maybe not. So how far are you with your AI from building a little robot machine that can help with pollination, for example, in places of the world where we've unfortunately killed the bees?
Yeah, well, so unfortunately, the pollination problem, I think, is much more a mechanical problem than a brain problem. So if you put if you had a pollinator robot that was adept in flying around reliably, and you put them right into a field of to-be pollinated flowers, I'm sure they wouldn't have too much of a kind of computational problem to do that. It's about the dexterity of the insect and so on. All of that being said, we're doing some research into navigation where we're actually using bees as an inspiration directly to build robots that navigate on the same principles. And these robots we have, and they're driving around in the lab. We're just taking them outside.
That is so cool.